Hello guys, welcome to Biology Made Easy. My name is Miss Natasha and I'm going to take you through today's topic. So today we are going to look at enzymes. We are going to define what enzymes are and we are going to look at how enzymes work. We are also going to look at the factors that affect the rate of enzyme catalyzed reactions. So without wasting any time, let's jump right into it. So enzymes are proteins that function as biological catalysts, and by definition, catalysts are substances that increase the rate of a chemical reaction and are not changed by the reaction. So basically, catalysts increase the rate of a chemical reaction and they are not changed by the reaction. The chemical reactions that take place in living organisms are termed metabolic reactions. So all the reactions that are happening in our bodies right now are called metabolic reactions, and almost every metabolic reaction is controlled by catalysts that are known as enzymes. So enzymes control the metabolic reactions that happen in living organisms. And an example of an enzyme that is found in saliva is amylase. So some people call it salivary amylase. A substrate is the substance that is present at the beginning of the reaction which the enzymes work on. So basically, a substrate is a reactant, and the product is the substance that is produced as a result of the reaction. The active site is a dent in an enzyme that has a complementary shape to a substrate. This is the site where the substrate binds onto the enzyme, forming an enzyme substrate complex. So the active site is the site where the substrate binds onto an enzyme, and when the enzyme binds onto the substrate, a complex that is known as the enzyme substrate complex is formed. Moving on, let's now look at how these enzymes work. This is also known as the mechanism of enzyme action. An enzyme works by allowing the molecule of the substrate on which it is acting to fit into it. So basically, an enzyme works by allowing the substrate to fit into its active site. So the active site is the site at which the substrate binds onto the enzyme. So the fit has to be perfect and specific. So this mechanism is known as the lock and key mechanism. The enzyme is the lock and the substrate is the key. The enzyme is the lock into which the substrate molecule fits, and the substrate is the key which fits into the lock. So the shape of the enzyme and the substrate are complementary to each other. This means that they fit perfectly into each other. For example, amylase is complementary and specific to its substrate, starch. All enzymes have active sites that exactly fit the substrate, and each enzyme can only act on a particular substrate. This means that each enzyme is specific to only one substrate and it can only catalyze a single reaction. Previously, we talked about the lock and key hypothesis or the lock and key mechanism, and we mentioned that the enzyme is the lock and the substrate is the key. So there is only one key for the lock, meaning that the enzyme can only catalyze a single reaction, or it is only specific to a single substrate. For example, amylase cannot act on proteins because starch is the substrate for amylase. So this diagram illustrates the lock and key mechanism. The enzyme is the lock and the substrate is the key. So the substrate molecule has a complementary shape to the enzyme and it can fit into it like a key. So this is the enzyme and this is the active site of the enzyme. So the substrate fits perfectly into the active site of the enzyme. So this is when catalysis takes place, and these are the products. The enzyme changes the substrate into new molecules called products. So the enzyme remains unchanged at the end of the reaction. And by definition, catalysts are substances that facilitate chemical reactions, and they remain unchanged at the end of the reaction. So the enzyme is a catalyst. It remains unchanged at the end of the reaction. Let's now look at the properties of enzymes. So enzymes are proteins. They are catalysts, meaning they are not changed by a reaction. Enzymes are specific. They catalyze only a single reaction. They are specific to a single substrate. Enzymes are inactivated at high temperatures, meaning they are denatured by heat. Enzymes work best at a particular temperature, which is known as the optimum temperature. And enzymes work best at a particular pH, which is known as the optimum pH. So the factors that affect the rate of enzyme catalyzed reactions are temperature and pH. The first one we are going to look at is temperature. So at high temperatures, enzymes are denatured or they are damaged. At high temperatures, the enzyme molecule loses its shape to an extent that the active site will not fit perfectly with the substrate. So at this point, the enzyme is denatured and it will no longer catalyze any reaction. And at low temperatures, enzymes are inactivated. So at high temperatures, enzymes are denatured, and at low temperatures, enzymes are inactivated. So enzymes work best at particular temperatures, and these temperatures are different. So the temperature at which an enzyme works best at is known as its optimum temperature. For example, MLS works best at around 37 degrees Celsius. This means that the optimum temperature for MLS is 37 degrees Celsius. And most plant enzymes work best between 28 and 30 degrees Celsius. This means that the optimum temperature for most plant enzymes is found between 28 and 30 degrees Celsius. This graph shows us how temperature affects enzyme activity. So at low temperatures, the rate of the reaction is low. This is because enzymes are inactivated by low temperatures. And as the temperature increases towards the optimum temperature, the rate of the reaction increases. So in this graph, we can see that the optimum temperature for this enzyme is 40 degrees Celsius. So as the temperature approaches 40 degrees Celsius, the rate of the reaction increases. But as the temperature goes beyond 40 degrees Celsius or the optimum temperature, the rate of the reaction decreases. This is because at temperatures above the optimum temperature, the enzymes are denatured. And when the enzymes are denatured or damaged, they lose their shape. And when they lose their shape, the active site will no longer fit perfectly with the substrate, meaning catalysis can no longer take place. So at temperatures above the optimum temperature, enzymes are denatured and the rate of an enzyme catalyzed reaction decreases. The pH of a solution represents how acidic or how alkaline the solution is. The pH at which an enzyme works best at is called the optimum pH. And if the pH deviates from the optimum pH, whether it goes above the optimum pH or below the optimum pH, the enzyme is denatured. And when the enzyme is denatured, the active site is disrupted and it will not fit perfectly with the substrate, meaning catalysis will not take place. So the pH of a solution affects the shape of the enzyme. So pepsin works best under acidic conditions or at low pH levels, and amylase works best under alkaline conditions or at high pH levels. So basically, the pH at which an enzyme works best at is called the optimum pH. This graph shows us how pH affects enzyme activity. So pepsin, which is a protease in the stomach, works best under acidic conditions or at low pH levels. So as the pH increases, the rate of the reaction decreases, meaning we have deviated from the optimum pH, which is acidic. And most enzymes work best at pH levels of around 7 or at a neutral pH. So if we increase the pH, the rate of the reaction decreases. And if we decrease the pH, the rate of the reaction also decreases. So basically, if we deviate from the optimum pH, the rate of the reaction decreases. So we have come to the end of our session, and in today's session we looked at enzymes. We defined what an enzyme is, we looked at how enzymes work, and we looked at the factors that affect enzyme catalyzed reactions. So if you want to research further on this topic, I highly recommend that you read the biology course book. So thank you guys for watching until the end.